All right, welcome back. We're at episode 2 now. We have a name of our podcast, Go with the Flow, a cytometry podcast, right, Christian? And we have a special guest, Ben McLeod, coming to us from Canada. He's the founder of Convey Bio and the co-host of the Biotech Voyager. So we'll let him explain a little bit more about what you're doing and what's going on with those two things you're undertaking, Ben.
Well, thank you for having me. Yeah, I guess I'm a scientist by training, turned somewhat marketer and content strategist for biotech. When you've got very complex science, there's a lot of really difficult ways and ineffective ways that you can try to share that science on social media. So my goal is just to basically come in and help these different biotech companies effectively share their science on social media, primarily by using the medium of their executives or their founders, since people just tend to trust a face more than they trust a random logo, and that tends to be a really effective strategy. So that's mostly of what I'm doing on at Convey Bio is basically providing those content strategy skills and services to biotech companies, and then separately, yeah, working on the Biotech Voyager, which we have the rather audacious goal of becoming the sports center for early stage biotech. And it's been a lot of fun so far.
Awesome. Do you think for the Biotech Voyager, just curious, do you think that you'll like have is the goal to have other people like they have ESPN reporters and stuff go out to like a game or something? Like, do you think you'll have different people go out to maybe like the start of a clinical trial or something like that and kind of in the real juiciness of what's going on?
That is one of the ideas, pretty far down the road. For right now, it's myself and my co-host, and we'll probably be the guys who are going to do that initially. But yeah, we have discussed this already. Is would we be eventually be able to have like on the ground reporters who are coming back to us and reporting in from a conference that we can't make it to, or they're on a client site somewhere, and we kind of get to do a site tour virtually or whatnot with them actually there. So yes, that would be the long-term goal, but a lot of things have to go right before that's going to take place.
That would be cool. That would be very cool. I have, yeah, another building off of that. Like, have you guys seen this? I saw it this week, I think, on LinkedIn that I think it's Kalshi or some other betting app is starting to put in betting, like sports betting, but on clinical trials. Did you guys
Wait,
see
what?
that? Yeah.
No.
No.
Yeah. I forget which app it is, but it's one of the apps out there. I don't want to speak out of turn, but one of the apps is starting to put in like betting on the clinical trial outcomes.
I don't know why,
Oh,
but that makes me very uncomfortable.
Yeah. Yeah. Like, what do you think about that? Yeah. Kalshi. That's it. Yep.
I mean, I think in general, gambling isn't a great idea. There's probably more effective ways that you could use your money, but I wouldn't say I have a strong opinion one way or the other. Aside from, for the most part, I try not to gamble myself. Seems like a difficult path to go down, and you could land yourself in the hole pretty quickly. And then, if
Yeah.
you're also adding in the ethical side of it, being like, I really hope I make some money if, little kids don't get better, might
Right?
Exactly.
might
Because
get problematic quickly.
yeah, because like yeah, if you're betting that it's gonna work out, more power to you. But if you're betting against it, because with any bet you have to bet it for or against, right? That like Ben said, that makes me uncomfortable.
Yeah, it feels a bit unethical. When I first saw it though, I was thinking like kind of I can see the obvious differences, but is it all that much different from like venture funds choosing which program gets funded? Like that's sort of betting in a way. It's obviously not exactly the same, but and you need that money to fund development, right? But this is like you're not getting and none of that money is going to the companies to help keep things going or any of that. So it's like it seems unscrupulous in my opinion.
That, and right, it's probably going to be really difficult to track insider trading, right? If,
Right.
I'm a scientist at that company, and maybe it's a little bit too easy for me, but then I like go to my mom's wife's cousin and say, you should make a bet on some insider information I have, and, I'd like a really nice birthday present.
Exactly. Exactly. Yeah, or even like trial site people, like personnel working at a site, like what if they mess a sample up or something like that, or there's a lot of issues with it.
Yeah.
Yeah, I don't know.
Yeah. It's crazy though. I sent the link in the chat so you guys can look at it
Oh,
after this, but
appreciate that.
yeah, it's wild.
Yesterday, just changing gears a little bit. Yesterday, Eric and I we were chatting a bit about Aesop's new release with their new drug. I don't know if you've seen it yet, Ben. The
No.
press release they put out on, believe it was the 20th. I want to say that it was the 20th, but I found their post quite intriguing, being the first of its kind. Let me share the news one really quick. Since you mentioned that you guys enjoy being kind of like on the forefront of what's happening in CG, let me find it real briefly. But what's some of the new innovations that has your interest piqued, Ben?
Yeah, I think there's a couple different things. One of the really big things inside of cell therapy, obviously, right now is in vivo CAR T. I think it's probably a little bit overhyped, just because, again, to some extent, it's going to be nice to actually get some more clinical data and readouts from that. And realistically, we're not going to have any of that really rich data for at least several years. But then the other thing that I do find really fascinating, just kind of with more standard autologous manufacturing, is the it is like how quickly the automation and the scalability components are kind of starting to come together. There's been some really exciting news just kind of even over the last couple, like so far this year, right? In terms of how can we get cell therapies away from this being a really manual, labor intensive process to something that's much more equivalent to an automotive assembly line, where you've just got this really precise set of machines that are working. You really reduce the chances of operator error or human contamination. So that's one of the big things I'm just kind of seeing this year. Like the automation play is finally coming online, and it's long overdue. And then I don't know if you guys saw, I think it was a couple weeks ago now, Carl June's group had followed a cohort of patients who had received CAR T, and they now have like a 10-year bolus of data of like following these folks. And it's like, if you get to 5.4 years in the particular cancer and CAR T that they're looking at, you're clear. You're not getting this again. So we're getting closer and closer as well to like CAR T therapies, at least in that particular circumstance. And obviously more data will come out with time, but we're just getting closer and closer to functional cures within the cell therapy space, which is just really awesome to see.
I mean, just it's interesting you also bring up Carl June because there's Emily, right? The first pediatric getting it, and now she's at Duke University pursuing her degree, and she got CAR
UPenn.
T when she
Yeah.
was a little girl, right? And now a successful scholar. So I think, yes, I think the good news are on the horizon, but not to be a Debbie Downer, but I think the education of the general public is still severely lacking, as well as access to reimbursement of the cost for therapies. You talk to most people, I would say on the average, people don't know what a CAR T is. They don't know immunotherapy is. They don't know that there is now a living therapy or a living drug that could potentially cure your cancer, provided it's one of the allotted ones, right? Like maybe a TL leukemia or something. But it just seems that people are still very unaware of it. What are you guys' takes on that? Do you think we're doing as a community enough to generate education, or wh- where do you think where do you think the ball is getting dropped, and how do you foresee that this could be improved upon? Because we're hearing a lot of different CAR Ts being developed now for infectious disease, autoimmune disease, metabolic diseases. So, but again, the vast majority of people are not getting it. I read an article not too long ago that less than 20% of the people eligible, not even the people that could potentially seek it out, but people eligible are the ones receiving treatment. So I don't know. I
Wow.
wanted to pick you guys' brain on this.
That's insane. I didn't realize it was that low. I think
It's super low.
I mean, it
Yeah.
checks out when I tell people, because I do a lot of CAR T analysis and things like that. And when I tell people that, I try to gauge what level of like science understanding they are, and then gauge what I say based on that. I think honestly, like COVID kind of helped things because it made immunology a little bit more in the zeitgeist of like, oh, they take mRNA and they express a protein in you to like give you a certain functional response. So, and like there's a lot of talk about T cells. So I think some people, and I could be biased, I'm in Boston, which like there's a lot of pretty well educated people here, honestly. But I think some people are starting to understand that, like, oh, there's these other aspects of our body that we didn't know about. Like, people know about the heart and the lungs and your muscles and your bones and skin and all that. But, like, cells aren't really talked about in like high school biology class as much, right, as the whole systems. So I think starting there and like starting to put some more cellular type education in earlier in the school system could be a good way to do it. But I think I try and tell people it's your immune cells, like T cells, that fight off various invaders in your body. We take them out, re-engineer them to go see a protein, and then they can go and attack the cancer. So something like that, but it's definitely isn't a big educational problem. It's hard to tell people that are used to, taking a Tylenol like it's a little pill. You mean there's other drugs that aren't a pill? That's kind of crazy. Like, what do you mean by that? So I think, I don't know, it's an interesting point and definitely needs some refinement, but yeah, I don't know. I don't have a solid answer.
Yeah, I think so. I recently, earlier this summer, I got to help with an event called Shortening the Brain to Vein Time at the Solaris facility, and Tom and Emily Whitehead were there. So that was really cool. Got to meet Emily for the first time in my life. And one of the things that was brought up at that event that dovetails really well with this is we often obsess over, okay, can we take the amount of time that a CAR T is required to get made from like 14 days down to maybe like 7 days, maybe from 7 days down to 2 days? Or could we even eventually get to a point where like it's just occurring in the same hospital where the person is getting treated? Do you know what's way worse is the amount of time that goes between a person's first diagnosis and them becoming aware that CAR T therapies exist. And it's something like 160 to 180 days. So we obsess over, okay, can we really reduce this amount of time that's required to get a person the treatment? But realistically, the biggest challenge right now is just making people aware of it earlier. And one of the big barriers to this is actually physician awareness. We kind of assume that all these different medical doctors are aware that cell therapies are at the stage they're at, and yet they're not. The industry, the advanced therapy industry, has done a really poor job of educating the physicians and going to the physician conferences. And we're just kind of assuming that they're kind of keeping up to date with all of our science and everything that's going on. They're not. They're busy treating patients. And they kind of still view a lot of these different cell therapies as experimental, as something that's not really a good fit for their patients. And so something that Tom Whitehead actually talked about is the fact that now what's actually happening more and more is patients themselves are going off and they're talking with AI, or they're doing research themselves, and they're coming to their doctors with, hey, there's a clinical trial for my kind of cancer inside of the cell therapy space. I'd like to be a part of this, please. And the doctors are kind of like this middleman that aren't quite as up to date and don't have the same level of motivation as like the therapy developers and the patients. So there's a bit of a gap there. It's crazy.
That's a great point and good gap to identify. I think it really hits it like how overworked a lot of doctors are, especially when they're doing clinical duties. And I think it's a good point too of like us getting out of our bubble a little bit to make it easier for these doctors to know what's available and what's up to date. Like we're all used to working with people that work in this field and are very up to date, right? So it's hard to remember sometimes that there's the average. Like most
Yeah.
of what we do is probably people at the cutting edge of science, but there's the average people that could be great doctors but just don't have the bandwidth to keep up. So yeah, I think that's why what you're doing honestly is super important. Like, how can we get it in front of more eyeballs that are actually on the ground doing medicine? That would be really helpful, I think.
Well, not only that, but I think I would also say not necessarily just the specialist, right? But I would even posit that we need, as a community, we need to also provide some degree of education to PCPs, because the primary care physicians are the ones that generally I mean, I don't know how it works in Canada, Ben, but here in the US,
It's worse.
some insurance, right, carriers require that your PCP give you a referral and authorization to go see someone else. So if this PCP doesn't even know that CAR Ts are a thing, and believe you me, I've met quite a few of those. How can you possibly how can the patient know that that's an available therapy, right? Should it become necessary, rather than when you're flustered and you just receive a diagnosis that you may have a type of cancer. At that point, you're not considering of doing research, but you're panicking about what are the next steps to secure your loved ones, getting all your affairs in order, how you're going to pay for all of this. It becomes an overwhelming time. And I think that's when you need your PCPs and other professionals to come in and hold that patient's hand and say, don't worry, there are treatments or there are clinical trials, rather than putting the onus on the patient to go do that research. Much like if you go to an infectious disease doctor or a migraine doctor, where you see those posters that has all the various therapies available and the drugs, And, while you're waiting for your doctor to come in, people, have nothing else to do in that waiting room. So they look at the posters and start reading a little bit. I'm still surprised that that's not posted in PCP's offices or at the very least in oncologist offices, right? You'd expect that in an oncology office, in the waiting room, there'd be magazines about CAR T or some advertisement on that. Inside the waiting rooms, that there'd be posters about it. When I traveled to China, I remember I was in Shanghai in one of the hospitals that does a lot of CAR T. At the time, they were doing 7 clinical trials and they were plastered throughout the hospital. And people were very aware that this was available to them should they be an eligible candidate. I don't
Right.
think we do that here. I don't know how it is in Canada. So maybe you could also shed some light on that, right? Because it seems like every nation has its own flavor of how they're approaching this. And here in the States, even every state, like Eric says, maybe in Boston or Massachusetts, people have better access. But what about people in the Midwest, like Wyoming or something? So I don't know. It just seems like there's a lot of gaps.
No, unfortunately, it's not any better up in Canada because we have like a governmental healthcare system, but the process of getting therapies approved and paid for through that health system is extremely laborious and slow to the extent that we don't have any approved CAR T therapies in the healthcare system yet.
Wait, there's
Wow.
no approved
They
therapy, even
haven't
as a third
taken
line of defense?
they haven't taken reciprocity with the FDA on these.
Well, it's one thing for reciprocity. It's another thing to then get it approved as a payable therapy inside of the whole
Mm,
Wait,
healthcare system.
so there's absolutely zero reimbursement, federal
That's my
reimbursement.
understanding. Yeah.
Oh, wow. And I'm over
So you
here
have to go to.
complaining in the US that we have minimal. We have some.
it's better than none. Yeah.
I did not read because
Wow.
usually
Good
hear
job,
that, right?
guys. There are some things that are better. More than
In
you
the
think.
healthcare system, because almost
Yeah.
everyone crosses the border to go buy their meds in Canada. And now I'm like, well, maybe it's not the holy grail we all thought it was.
It isn't. What becomes very inefficient and very, very slow and sometimes doesn't make any sense. So
When
yeah.
when you said that you met Emily and her dad, did you by any chance speak with, or I'm sorry, did you meet George, their head of the Emily Whitehead Foundation?
I have met him before. He wasn't there. But yeah,
Okay.
great guy. Yeah.
Yeah, great guy.
Unfortunately, gentlemen, I do have to jump, but
Oh, that's right. It's 10:30.
yeah, fantastic. Thank you so much for having me. Really, really excited to see where you go with this. I think it's very much needed.
Thanks,
Absolutely.
man. Thanks
Thanks
for
for
joining
coming,
us today.
Ben. Yeah.
Cheers. All right. See you later.
Bye.
Talking about CAR Ts and the access thing is crazy and the education is clearly needed. And I think especially as things progress, right? Like I think we're starting to see some things go earlier, CAR Ts being used earlier in the process, which like Ben was talking about 180 days for a patient to even learn that it's an available option for them is insane. And that needs to change. But I think also the
Yeah.
thing too that needs to change, quite frankly, I'm like very passionate about this, is CAR Ts get put like after third remission or third
Yeah.
recurrence of
Third
cancer typically.
a third line of defense. Yeah. So after everything else has failed, they approve it except Yescarta. I'm not sure about Kymriah. Yescarta has been approved as a second line of defense.
Right. So yeah, they're starting to move the stage up when these are actually deployed. But I still think it should be earlier, like right? Like this actually
Oh,
was a recent paper that I was involved with from City of Hope looking at giving CAR T cells after first remission. So basically patients diagnosed, they go in, they get immunotherapy and different kinds of therapies to get their cancer in remission, their leukemia, first remission, and then right away, without waiting for a recurrence, they take their cells, make CAR T cells, and then give them the CARs. And of course, like as you would expect, when you put healthier CARs or healthier T cells through the CAR processing, you end up with healthier CAR T cells that are able to go and have their effect and adapt and do better work in what they're designed to do than when you wait till someone's been through 3 rounds of radiation and chemo and all these things that they damage the cells. They're supposed to go after cancer cells, but we all know that they also damage healthy cells. So we looked at what happens if you give CAR Ts up front early on in the course of the disease versus later after 3 rounds, and it turns out that they work a lot better. Who would have thought? So that was a really fun project to be a part of just because of the implications that it has, right? Like it's the first time it's been given after first remission, and it's amazing. It's an amazing opportunity.
well, I think that's one of the added benefits, right, that people seek regarding, unfortunately, now the not so vogue idea of not a homologous CAR Ts, but rather allogeneic CAR Ts, right? That you're going to be getting them from healthy subjects or healthy
Mm,
donors where the cells are very healthy and they're going to do their thing. I agree with you. I don't see why, especially with the current data, the publication that it's now been 10 years, there's enough data to suggest that this is fine. Yes, there are side effects, but what drug has no side effect, right?
right.
So if I don't know. If you were telling me, hey, we rather get chemo where, non-specifically we're damaging all your cells and I'm going to lose all the amazing locks I have, or get CAR T, maybe Yescarta or Kymriah, and, yeah, there's side effects, but they're minimal and show up in a small percentage of the population. And the earlier I get, the higher likelihood it's going to succeed, and I'm going to go under complete remission. I think that's a no-brainer.
Right.
So it's just the biology supports the idea, right? But I think where we're getting caught up on is, well, it's so much cheaper to pay for chemo than it is to pay for a CAR T. So
Exactly.
we go down
Which
to
is,
resources, which kind of sucks
yeah,
because at that point, when people say, oh, every life is precious, every life is invaluable, I don't think that's how accounting is seeing it or the government finance offices. They're actively putting a dollar value, saying, well, you first need to use up all these lines of treatment that we know are potentially less efficacious now or less, let's call it accurate, if you will, than this
it's
other line of
cheap.
medicine, but it's a tenth of the cost, right?
Right.
So,
Yeah, and even that, like that logic is it's understandable. I don't agree with it. If there's something that works better, let's use that. And also, it kind of doesn't even hold up, right? Like if you give someone 3 rounds of chemo, let's say over 3 years, it knocks their cancer down for half a year, maybe 6 months of treatment, knocks it down for half a year. 6 months later, they're back on. Let's say they're out of work most of that time, right? Even if you look at which I again don't think is a great way to look at people, but if you look at people like the economy does, and someone that's off work for 3 years, well, they're not really producing anything for the economy, right? So they kind of you're losing a lot more, and that's just like the financial side of things, not to mention the patient and the patient's families going under chemo, getting remission. It's exciting, and then oh, it's back. It recurs, and then say that process is just takes such an emotional toll on people and mental toll. It's a lot. And so I think we would save a lot of strife and probably a lot of money if we keep moving CAR Ts further up front. Because we know like we were just talking about, they effectively cure a lot of leukemias at this point. Like
yes.
sure, there's some recurrences and it doesn't work for everyone, but much better than any other treatment available in pretty much any other disease state. I would argue we have something that actually can like pretty much cure a lot of people, which is amazing.
Yes, and I think to our earlier conversation with Ben, I'm sorry, is that I think if people knew about this, they would start demanding it more. And maybe it's my tinfoil hat, right, that I'm putting on, conspiracy theory, that I think the lack of education is purposely done so that population doesn't demand it. Because God knows if I have a family member, I'm going to try to get that family member or loved one. I mean, they're not mutually exclusive, right? But my loved one, the best treatment possible. I'm not
Absolutely.
going to be considering how much does it cost. If that could cure that my loved one from cancer, why would I not pursue that?
Right. Exactly.
So, or how can my loved one and I work together to so that my loved one becomes eligible for a clinical trial or receive a
Mm,
treatment, right? They're removing that opportunity and choice from the general population, I think, by not making them aware of it. And the reason why I brought up George earlier is I think he's doing an amazing job, and huge shout out to the Emily Whitehead Foundation because one, they raise funds to provide free treatment to children. Two, with CAR Ts, right? And two, they're very, very active on the Hill at the moment to try to increase reimbursement, increase access, increase education, and get government policies more in line where our field and industry is. And it's a huge massive undertaking. And I would love to be able to pick George's brain and see where that's at and what are next steps. But I don't know. It just seems that I think we need to just do better in from all ends, right? Because we can't just squarely place the blame on outsiders because
Right.
all of us are within the community. We have a voice and we should choose and elect to use that voice. And if we have any type of platform, try to get the information out there.
Yeah, I agree completely. I don't know if I agree fully though I agree with getting everything out there, and it's a lot of it should fall on the community to educate the masses, really. The hard part too, quick aside, is how much the public's trust in science generally has been eroded, right? Like we've talked about this before quite a bit of how now it's kind of like 50% of the country, and this is speaking more from the US perspective, like you 50% of the country that doesn't trust science at all, thinks it's all full of kooks and quacks and all this stuff. But then you have another 50% that do trust science, maybe some of them too much, honestly. But yeah, I think probably you're right in terms of the insurance companies certainly are advocating for education. They don't want to have to pay more. They don't really care that much if you live or die. Like they just want to make their money. I don't know if the industry as a whole is not advocating for it, right? Because it's a beneficial if more people know about it, they're going to sell more car Ts, which is good for the industry. So I think it's kind of like I see it more as obviously insurance doesn't want to cover it, and the industry a lot of times, and like we're both scientists and we know scientists and we all kind of act similarly, we get stuck in our own heads sometimes and stuck in our own little bubble of like we spend all day on this, of course everyone knows about this, right? Like how could you not? You don't follow the news or anything, you don't really listen, pay attention to these things that could cure you, affect you. So I think like stepping outside of that framework is helpful too, right? And I wonder if
It
there's
is.
something you could do as Supernova with your education platform towards like maybe working with the Whitehead Foundation and helping increase education to the public.
That is a very good point. I mean, I have reached out to George and we're meant to have a meeting and discuss it further. He's just a very busy man.
I can
Um,
imagine.
but I do need to reach back out again and get that going because it is near and dear to me, right? That I can't just be pointing fingers and not doing anything myself, right? Like the
Right. You
the old
can't
Chinese
call out a
saying.
problem and not have a solution.
Exactly, because at that point, I'm just whining and complaining. But I also agree with you. It's like a lot of times we get stuck in our own little bubble with other immunologists, other flow cytometrists, and we just continue saying, this is horrible, but we're not really talking to anyone outside our bubble. Or only if it's convenient do we talk to other people outside our bubble. And we might need to change that, I think. But
Right.
I think that it's a good segue, Eric, when we were talking about side effects, education, and who is eligible and all, the myriad of topics we've been discussing, I wanted to briefly talk about a paper that, which congratulations, the UN, I think it was City of Hope, or correct me if I'm mistaken,
Yes.
put out regarding a clinical trial, phase 1, for geriatric patients. I read it. It was a really cool article. I had some questions that I'm hoping you could address.
Sure. I'll do my best.
One is with people that are over 64, we know that the thymus is no longer producing the same amount of RTs, recent thymic immigrants, as someone that is under 55, right? Average
Hmm.
55, when the thymus starts generating a lot less TREs. So when so you now have this circulating bag of T cells that are most likely antigen experienced. So when you modify them, I saw in your report that there were significant immunoinflammation phenotypes. My question is, how is it that, or how do you guys know that this was due to the circulating or the presence of any cancer related epitope being targeted by the CAR Ts and not an innate state of where these cells were at prior to modification? Because even through modification, you're already expanding them, activating them. So you're driving them further than they were already, right, in that person's body who's already on average 64 years old. So, how is it that you guys attributed the pro inflammatory phenotype exclusively to this rather than saying it could have played a part, but we also say that it has to do to the age of the subjects, and prior antigen expression, prior antigen interaction where it may have led to memory.
Right. That's a good point. I think the big answer to that would be that these are matched groups so that the cohort demographics for we have the complete remission one and the relapse refractory patients, they're matched. So the idea that if there's something that's more age attributed, and obviously it's a small sample set too. It's not a huge like big phase 3 trial. So there's not like hundreds of samples. We have, I think, 16 and 16, and they're matched, so we assume that they come from a similar pool of T cells. Obviously everyone is different, I know every CAR T is different, so it's not necessarily that it's an age related phenomenon, especially because we're comparing both the pre-infusion product and the post-infusion products in both groups, and we see these consistent throughout. So it may be that led us to believe, and again, I don't think the paper fully supports this and says this yet because we can't definitively prove it, but it does indicate that after multiple rounds of normal traditional therapies, the T cells come in more inflamed, which makes sense biologically, right? And less adaptable to their environments, right? So that's kind of what we saw in the paper. And there's a ton of data we have that wasn't even put into the paper, honestly, that shows and kind of backs this up, that this is a pretty consistent signature that we see, where basically the relapsed refractory patients had, like you said, more inflammation, like more of those programs showing up, at least is from what we could see in our data. And also the bigger thing too, or potentially equally interesting, was the metabolic profiles of these patients. Effectively, we found that the earlier you gave the CARs in the complete remission, the CARs were able to have more metabolic plasticity effectively and engage in different metabolic pathways to generate energy to be able to go and become effectors and go have their effect and kill the cancer cells. So it's more, we think right now, based on the data that we have and we show in this paper, that it's really a result of these patients have healthier T cells because they haven't undergone all these treatments.
I see. Another really cool thing that I saw from the article that I don't see much with the phase 1 clinical trials or even phase 2 for that matter is that you guys did quite a bit of various techniques looking at basically a multi-omics type of assay where you saw gene expression for metabolics, you looked at proteomics, presumably flow, to look at, well, the protein expression, right? So I thought that was really neat. Can you shed some light as to what drove you guys to do these type of techniques that are not the norm for a phase 1 clinical assay?
For sure. So this is kind of an approach that I've helped develop with Dr. Leor Goldberg at City of Hope. He led
Mhm.
the kind of exploratory aspect of this paper, and it's it's really something that we like to do, and we do this in the analysis that we perform too, is look at the kind of same samples or same type of things coming from patients. Like in this case, we use Cytometry to look at the CAR T cells directly. Then we used mass spectrometry to do this untargeted metabolic profiling, right? And then we also use Seahorse, which is a metabolic assay that looks at how much production of different pathways is going on in the cells. And so we take these multiple pronged approach, and like you said, it's like a multi-omic kind of thing to try and tie it all together and see if we look at the same samples at different angles, do we see the same thing or do we see differences between them? And obviously each thing shows slight differences, but overall they kind of converge. And I think we do a good job of setting up our hypothesis testing and having good experimental design so that we kind of know what to expect, right? Like usually we'll do the Cytol first because it's kind of the quickest and most easiest one to do out of the three. And we see that we have this signature. That's interesting. And we'll set it aside. And then we go to the mass spec data and we look at that and we see we're seeing the same pathways showing up here that we saw on the Cytoff. And then we look at the Seahorse and we see what that says. And sometimes we see different things. So like in this, we saw that again, like I talked about, the CAR Ts from the CR1 group have kind of more plasticity and more spare respiratory capacity, which is one of the measurements that the seahorse assay gives you. So that kind of indicates that they're a little bit more fit, right? Like if you think talking about educating the masses here, like if you think about a high performance athlete, everybody knows they're going to have more lung capacity and more spare respiratory capacity, like at a systems level. Cells kind of have the same thing. They need to burn energy to be able to do the cellular processes. And so having extra respiratory capacity in a cell is a similar kind of thing where it has more ability to go and quickly shuttle different pathways to engage and produce energy to be able to go engage the target, in this case the cancer cells. So yeah, we generally look at, like I said, just these different angles, and we see what converges between them, and that gives us a better ground to stand on to say that we see this signature in this group versus this group. And in this case, that's what kind of led us to the notion that these cells that are, let's say, fresher, newer cells in the CR1 group have less inflammation. They have more metabolic capacity and functional fitness, basically, to go in and do their task.
I see. I also had a question, and this is more on my own biases and preferences. I believe you guys use high dimensional analysis, correct?
Yes. Which figure?
That's where for some reason I could have sworn I saw that you guys use FITSNI and compare pre and post,
Oh,
but
in this
maybe I'm
paper,
confusing it with another paper.
yeah, I don't think we did in this paper. We kind of took a more basic approach on the Cytoph analysis here.
Yeah, I'm sorry. I was confusing it with another paper.
No, all good. We had another one this year too that we did use some UMAPs in. We compared some things.
Okay. So, but you guys did decide to kill the cells with Cytol. You guys did decide to kill the cells with Cytol.
Yeah, we prefer to blow them up instead of bleaching them.
Uh,
It really just comes down to, honestly, it's so clean, the data, and with like CAR Ts being fairly limited in some of the samples, like some of a paper we did 3 years ago, something like that, we were looking at CAR T cells in the brain, like in the CSF, and so there
mhm.
weren't a ton. And if we're using flow, we're going to lose some by compensation and just generally, right? And of course, in the sampling of each of these different techniques, you're going to lose some cells, right? But in the Cytoph, like it really gives us a nice, clean population to be able to gate on and identify our CAR Ts. So it's really that plus, I mean, as you can see from the panel in this, like this, there's surface, there's intracellular, there's some phospho stuff. Like we can do all of that in one assay that's works very well in Cytoph and has some better resolution on some things. So that's really the rationale behind it.
Yeah, I mean the data that you guys presented it's very clean, right? And the I have been thinking again, we talked about it 2 weeks ago, but I'm still thinking about it and mulling it over in my head about current controls for compensation in spectral cytometry, especially now with the first clinical validated assay that we were talking about
Right.
Europe earlier.
I'm super
Um,
curious what that looks like.
yeah.
The controls of like what is the control regimen there?
Exactly. And I think if I remember correctly, you mentioned that when we were talking about this, or I can't remember, we were talking over the phone, that I mentioned that what's recommended, right? Pie in the sky, which is not practical or realistic for a lab, is to run the autofluorescence controls and remove the autofluorescence signatures from each single sample, right? So then and then you still need your FMOs, you still need your single color controls, blah, blah, blah. That at the end of the day, are we really saving significant amounts of money
And time
versus a
and
Cytoph
cells.
and time, where Cytoph, one of the biggest caveats that people argue against it is the cost.
Right.
But if we start talking that you have to have so many controls,
Right. Exactly. I mean, I've done this before of a cost comparison. And look, I obviously come from a Cytoph background, so I may be a little biased, but I work with all the different technologies all the time. So it's not like it's a hard and fast rule. I haven't been fully convinced on the economics of it between once you get to a super high parameter assay, right? Like if you're doing 25 colors, maybe even 30, spectral is probably going to be cheaper. Now, roughly at this point, like this was a few years ago that I did this study, but look, we looked at spectral versus Cytoph and calculated the cost, the data quality, these sorts of things of what's the break point where it makes more sense to run Cytoph versus flow. And it was about 15 markers. Once you hit 15 markers, you're making 15, 16 tubes for your single color controls if you're doing it properly, right? Like most people aren't going to do all that all the time. But if you're making your controls properly, you're going to end up with maybe 20 tubes, maybe more of controls for a 16 color panel. And then you have all your samples. So, and it's also, a lot of people like to use beads for single color controls and comp controls even sometimes, but we know that that can be problematic and it's not the ideal case. So if we're talking just straight ideal case versus ideal case, once you get beyond that, like Cytoph makes way more sense from an economic standpoint. You can stay in one tube, which you can also very easily, and I'm not trying to make this a case for Cytoph, but I do believe in it. And like, just like articulating my thoughts and feelings on it, you can make one tube and have all kinds of surface receptors, intracellular markers, phosphoproteins in one tube. It works well. Obviously, there's time to set up the assay, but that'd be true for either Spectral or Cytoph.
Right.
You don't need to do compensation or unmixing. There's none of that. Effectively, once you have your titration set, and let's assume you're not doing a bridging study, which we also talked about before, once you have your titration set, that marker, as long as you're standing it at the same concentration that you ran your titrations and your optimization of the panel on, it's going to give you the same output at the same like intensity level, essentially, if you have similar protein expression, obviously. So it's going to stay consistent. There's not like changing the voltages and having the flow plots look differently. There's none of that. There's a lot less parameters to tune and or mess up, honestly. So when you get into the high parameter space where it's super important that every marker is working well, it just
Yes.
becomes a lot easier. You don't have to do single color controls. You don't have to do all these 22s of controls. You should still have some controls in your data, right? Like a batch control at the very least. And arguably it's good to have just like a technical control to make sure your assay is working appropriately. But generally, once you get above really 20 markers, it really starts to make a lot more sense. Now, the access and the upfront costs can be scary to people.
I see. Yeah, I mean, the answers, I could be mistaken, but Cytophs now are rare and few in between, at least over here in the southeast of the US.
There's one at
That-
Moffitt I know of.
right.
I don't know if there's any elsewhere.
Exactly. So that's like one, not one, but like one facility that has
Right.
Cytoph for quite big region in the US. I'm sure it's a lot easier if you're in San Francisco or in Boston, but if you're not, then it's like, how do I get my samples done?
Yeah, fair point.
But I do agree that now that I'm thinking a lot more about this whole thing of autofluorescence, and I don't know why I got so hung up on it, but I have,
It's a
is
big problem.
it's a good problem, and I haven't seen someone that's given me an answer that is satisfactory, to be honest. But I think I don't think that's an issue with Cytoph. If you were looking at something where you're doing very high parameter, you're looking at very precious cells that you cannot afford to lose any signal, I am starting to and I'll say I'm starting to. I won't say I've converted, but I am starting to think that Cytoph might be a better option. And you know that when we hung out and chatted at the Cyto 2025, I was not the biggest fan of Cytoph. I was
Right.
like, it's an obsolete technology that kills cells. But I'm starting to come around. Yeah,
I mean,
I
it's just
think
about using the best tool.
exactly. Using the best tool for the best problem, right? And not closing our minds off and having a preference. I mean, we can still have a preference, but not being
Right.
blocked off to any other tool just because it poses, I don't know, competition to our preference.
Exactly. Yeah, and yeah, just definitely biases things. The way I see it, I think like how I kind of see the workflow, I think they all can work together, right? Let's take like a traditional drug development timeline. You're doing your discovery work, your pre-IND work, like just trying to see what's going on with your drug or your product. I think Cytoph is a great tool for that because you can cast a broad net, you can do it pretty easily. And like we were just talking about, like the cost difference isn't all that much when you actually calculate it and think about it. Maybe that's some good homework for me. Calculate, like if you're running a 40 marker panel and doing it right and having all the controls, what the differences are. But The early stages, Cytoph really excels because you can find a lot of information. The cost per tube is very cheap and it's great. Maybe even into phase 1, like we did here, where you can use it, do some biomarker discovery, do some more exploratory translational stuff. Once you get to phase 2, where you're having a lot more patients, maybe some people still are going to do Cytoph, and it's very reasonable to do so if you're still like mining for data and finding different like biomarker signatures, right? Once you get to large phase 2s and phase 3s and you like have refined what you're looking for, it makes a lot more sense to go with like a spectral or even maybe a traditional flow if you can condense the panel down enough. Like use your Cytoph, use the best tool available for your discovery exploratory work. Arguably it's Cytoph. Do that. Then you pare it down, see, because not you and I both know like not all the markers in your assay are gonna even really tell you anything in an assay. So then you can strip all the non-important things, find whatever signature you find, make a panel for that that's more specific, maybe like 20 markers, 15, even less, and then deploy that in later stages. Because it is a lot faster. This is probably one of the big hangups. Flow is a lot faster, like you're running at thousands of events per second, even tens of thousands, versus hundreds in Cytoph. So that's a big consideration. But if you already have a signature identified and you just want to churn through samples and see how well that holds up, then you can just use a spectral flow and do that, right? That's kind of how I see the pipeline. Early stages, go broad, go big, go Cytoph. Later stages, throughput is more important than breadth of the assay, right? Use the more refined approach.
I mean, I would agree with you the vast majority, but I would say post BLA, right? Post phase 3. So I would say that once you're doing immunomonitoring, like this 10-year paper or a 5-year follow-up, I think it behooves the researcher or the company to do a deep dive to see how a bunch of other markers, whether they be metabolic or whatnot, have been impacted by CAR T therapy. And I think that's when you bring back the big and broad panels, potentially Cytoph. I've been kind of proposing a new idea of a 3-tiered stage of how to do flow workflows within the regulatory space and what technologies are best suited, blah, blah, blah. And one of my proposals is that in tier 3, which is discovery, I put it, like you said, in R&D, but I also put it at the very end of immunomonitoring because that's the only time we're going to get a real glimpse of what is happening 5, 10 years later, right? And if we want to take a deep dive, then we need a huge panel at the very minimum, like 22 plus, right? And at that point, if we're talking about cost effectiveness and blah, 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 is it a good idea to bring back the Cytoph at that point? But my reservation would then be, what about comparability and tech transfer between the different technologies? How do we compare data from one to the other? So,
Yeah,
but
it's an issue.
it's an issue, but I do think that, like I said, I'm coming around to thinking that Cytoph has a place in CGT, whereas before I was kind of like this obsolete technology.
I mean, look at it this way. We were just talking about Carl June. He uses Cytoph, that 10-year response paper, Cytoph based. Because again, I think I don't know exactly. I haven't talked to him about why they use that, but the kind of like what I hit on. At 10 years, you're not going to have a lot of CAR Ts floating around, right? Like ideally you don't, because that means that if you do, they expanded and you have cancer again. Ideally, you're not going to have a lot of CAR Ts floating around in your blood. So to be able to detect that, like you want something that can detect rare events without potentially throwing some of them away due to pre-processing of the data. And that's really kind of where it shines. So yeah, that's an interesting approach to like bring it back at the end. Makes sense.
Yeah, and I think that's and that's exactly what we're talking about, right? Immunomonitoring. That's why I'm proposing that tier 3, because you brought it in the very beginning at R&D level to generate your hypothesis. Then I believe it's very appropriate to use conventional. I'm not sure if I would recommend spectral after ADPD. I think spectral sits very well in ADPD. I would not use it for QC, although now the jury's out with this new spectral IVD instrument. I cannot say pro or cons against it because I haven't reviewed it, but I am highly interested. But going back to this, I think I would say, yeah, definitely large panels, which either Cytoph or Spectral and Discovery. ADPD, most likely just conventional or spectral, and then QC, I would say stick with conventional just because it's tried and true and tested.
Right.
Clinical trials, probably I would say sticking with conventional because of all the validations you need to do. And you're not going to be running huge at that point. You're probably going to be running 12 to maximum 18 colors. Odds are less than 12. But then when you get into the immunomonitoring post licensure, I think we go back to large panels and bring back either the spectral or the cytol. And then or multiomics. Like I said, I really like what you guys involve about the metabolomics because that is, I don't think it's finally getting traction, but I think it's
Mhm.
largely overlooked despite how critical and crucial it is for our immune system.
100%. Yeah, that's why we've really kind of dug into this immunometabolism, as we say, of CAR T cells because it has been overlooked. Like the last now 3 papers that I've done with the City of Hope group has been focused a lot on the immunometabolism of the CARs because that's ultimately what matters, right? Like, and we've seen it. You send in a CAR that is hyper inflamed and highly, I guess I would say, immune homologous, right? Like they are on one path of or not immune, metabolic homologous, right? They're on one path, one like metabolic program basically. Like they're less likely to be able to adapt in different scenarios and environments that they find themselves in. And so like yeah, it's allowed us to figure that out, to figure that out. See that not only are these cells somewhat different in how they're trafficking through the body, but they're also different in how they're actually using energy to do their things. Like a cell that has a lot more energy, like if you eat breakfast and then you go out and like let's say you're working I had a meeting yesterday with some guy that came off his farm working. Like if you eat breakfast, you're going to be able to do a little bit more work on the farm than if you don't eat breakfast and then you're gassed by noon. So it's the same kind of thing here where the cells that are more able to eat their lunch, let's say, are able to go and have a better effect. And yeah, it's only through studying this in multiple angles, using a big, broad CyTOF panel that has various different markers in it, it allows us to see that. Because we saw there's no difference in their like subsets as we call them, because we basically transport the canonical T cell memory phenotypes over to CAR Ts, which is a little iffy if you've seen the data. Like they don't look the same. Like PBMC, CCR7, CD45RA plot, like it's beautiful. You get your nice 4 populations on that. Looks great. CAR T cells, they're like a mismatch of stuff. They don't necessarily have the same exact pattern and profile as normal T cells. So it's not in the subsets. It's in what they're actually doing inside that really counts.
Yeah, I agree with you. So thank you so much for you guys working on this pretty much, like we said, overlooked topic within CAR T. And just for the viewers, will you be posting the link to the article with the podcast?
For sure, yeah. I'll put the link to the article below this. It's from Blood Advances, so it's up. It's open source, and it'll be wherever the podcast is posted. It'll be there too.
Awesomeness. Well, Eric, I think we're a little bit over time, and thank you so much for, spending time together today and chatting up about flow and CGT and for Ben's commentary and his insights. So I guess we'll see everyone in 2 weeks.
Sounds good. Yeah. Thanks, Christian. As always, it's a great time chatting with you.
Likewise. Thanks, Eric.
All right. You're welcome. Bye, Christian. Thank you.
Bye.